Today we are going to discuss about intracellular receptors. So earlier we have discussed about the receptors which are found on the cell membrane itself. And today we'll look into the receptors which are by nature proteins, and these proteins are found in the cytosol or inside the nucleus of the cell. Now, small or hydrophobic chemical messengers can readily cross the membrane, and thus they can activate the receptor. For example, uh, the hydrophobic messengers, the steroids and thyroid hormones of animals. And hopefully, you have already studied the mechanism of hormone action. Now, this activated hormone receptor complex can act as a transcription factor, which in turn can um, switch on some genes. So, here in this diagram, you can see that uh, a hormone which was found on the outside of the cell, and the hormone receptor is found inside the cell. Now, what will happen as this kind of hormone can pass the lipid bilayer of the cell membrane? They can easily go inside the cell and can bind with the receptor protein itself, and thus they can form a hormone receptor complex. And this hormone receptor complex can go through the nuclear pore, and there they can act as an transcription factor. And the transcription factor can regulate the gene expression process inside the nucleus. And ultimately, the cell will generate the mRNA, and thus a new protein will form, and different cellular responses will happen due to this hormonal activity. Next, we will look into the next step of cell signaling mechanism, that is transduction. Now, it is a cascade of molecular interactions. So, mm, if you have seen the molecule, the relay race kind of thing, one is running with a with an object, and that object, he or she is passing to the next runner, and then again he or she is completing his turn, and then again someone will mm, someone will run. So it is a cyclic uh, order, and this kind of phenomena is happening inside the cell when signal transduction occurs. So it involves multiple steps, and during this process, a signal can amplify. So if one molecule is generated, so from that one molecule, 100 molecule can be generated and can be activated, and from that 100 molecule, 10,000 molecule can be activated. So it's a huge number actually. And this kind of multi-step pathways provide more opportunities for the coordination and regulation of different cellular activity. Now, these signal molecules, which are also known as relay molecules, which transmit the signal from the receptor to the response, are mostly proteins. The receptor, which actually it is a switch on and switch off kind of thing. So receptor activates another protein, which activates another, and so on until the protein producing the response is activated. So and at each step, the signal is transduced into a different form. Usually, a shape change occur inside the protein molecule. So many proteins are involved in this process. Two of these are most important. That is responsible for phosphorylation and dephosphorylation. In many signaling pathway, the signal is transmitted by a cascade of protein phosphorylations. So what do we mean by protein phosphorylation? It is addition of a phosphate group towards a protein. And generally protein kinase, we have seen in the last class also that protein kinase transfer phosphates from ATP to a protein, and that process is known as phosphorylation. Another protein is known as protein phosphatase, which can remove the phosphates from protein. So it is a reverse process. This process is called dephosphorylation. And these two processes, phosphorylation and dephosphorylation system, acts as a molecular switch, turning the cell signaling mechanism on and off as and when required. Here in this diagram, you can see that how a signaling molecule is bound to the receptor, and the second messenger, which is activated in this way, can activate the another molecule, which is inactive protein kinase. So that protein kinase can be activated, and then it will activate another protein kinase. So this is a chain reaction. You can see it's a long step. One by one molecules can be activated, and ultimately the effector molecule inside the cell will be activated, which ultimately generate the cellular response. So this pathway are termed as phosphorylation cascade. So the same way, the phosphorylation cascade can pass on from one molecule to another molecule, and it can generate lots of uh, branches in that pathway. So next important molecule are known as second messenger. So these are small molecules and ions, and the extracellular signal, which is known as ligand, that bind to receptor is a pathway's first messenger. You know, we have talked about the ligand receptor binding in the previous class, and these second messengers are actually small non-protein water soluble molecules or ions that spread throughout a cell by the diffusion process. So there is a difference between the first messenger and second messenger. So first messenger, usually doesn't pass the cell membrane. They can interact with the receptor molecule, whereas second messengers are non-protein in nature, and they are water soluble molecules. So second messengers participate in pathways which are initiated by the receptors like GPCRs, and receptor tyrosine kinase. The examples, notable examples are cyclic AMP and calcium ions. So we will look the 2-second messengers, examples of 2-second messengers. One is cyclic AMP. In short, it is called CAMP. It is most widely used second messenger, and the enzyme which is responsible for production of CAMP from the ATP molecule is known as adenylyl cyclase. So you can see in this reaction the ATP molecule. It is a structure of ATP molecule at the left hand side. The reaction is catalyzed by an enzyme adenylyl cyclase, which removes the phosphate, 2-pyrophosphate, 2-phosphate group from this ATP molecule, and thus one molecule of CAMP, cyclic AMP. Why it is known as cyclic AMP? Because it join with this ribose sugar. This phosphate joins with the ribose sugar by a bond, and that's why its nature is cyclic. So it is termed as cyclic adenosine monophosphate. So actually it is a AMP, but it is cyclic in nature. And another enzyme in next step phosphodiesterase, which can cleave this bond and it can produce the AMP molecule, adenosine monophosphate. Okay, so this is the reaction mechanism. Now, many signal molecules trigger the formation of this kind of second messenger, that is CAMP. Okay, now CAMP usually activates the protein kinase A. That is another protein. So, during this signal transduction mechanism, this second messenger activates another protein. So, these are the examples of that generalized mechanism, that CAMP is the second messenger which activates a protein, and that protein name is protein kinase A. The protein kinase A again can phosphorylate various other proteins. Now, further regulation of the cell metabolism, then which are provided by the G protein coupled receptor systems that inhibit adenylyl cyclase mechanism. So here is the pathway. You can see that when a first messenger, for example, the hormone epinephrine, can bind to this GPCR, and then the G protein is activated, and the activated G protein, with the help of adenylyl cyclase, can cleave the ATP molecule to form this CAMP molecule, which can act as second messenger, and that second messenger activates the protein kinase A, and that protein kinase in turn uh, in turn uh, generate the cellular responses. Okay. Another small second messenger is calcium ion, and other one is inositol triphosphate, IP3. So calcium ion is an important second messenger because cell can regulate its concentration. So there are many pathways how the calcium ion, concentration of calcium ion can be regulated inside the cell. Okay. Now, another signal relayed by signal transduction pathway may trigger an increase in the calcium in the cytosol, and then the pathways leading to the release of the calcium involve another molecule, that is inositol triphosphate. It is another example of second messenger, and diacyl glycerol, in short DAG. So these are also regulating the other pathways. So let us discuss about its about this pathway. So here it is. So first messenger bound to GPCR, then again that is activated G protein. So it activates the phospholipase C, which cleaves this molecule PIP2 into two components. One is DAG and another is IP3. So these two molecules can act as second messenger. So here the IP3 gated calcium channel is activated then, and calcium can release out from the endoplasmic reticulum towards the cytoplasm. So by this way, IP3 or DAG can be used as a second messenger molecule in the cell signaling pathway. Okay, so third one of cell signaling mechanism is response. So it is very easy to understand that cell signaling leads to regulation of the transcription or the cytoplasmic activity, and as a result of that, the ultimate cellular response will be generated. So it may be nuclear responses or it may be cytoplasmic responses. That depends time to time according to the cellular activities. Okay, so this we have already discussed, how the cell signaling is happening through the reception and transduction and then response mechanism. So this is a summarization of the three steps of cell signaling pathway: reception, transduction, and cell response. Obviously, this response must be fine tuned, and these four aspects of fine tuning must be considered by a cell. For example, amplification of the signal and thus the response. Sometimes few signal molecules are present, but the response must be um, response requirement is huge. So then that signal amplification is mandatory. It is required, very much required. Then the specificity of the response is also to be fine tuned. Overall efficiency of the response enhanced by the scaffolding proteins are also required. And obviously, the termination of the signal is also important phenomena, because I told you earlier class that if a if a cell signaling mechanism is turned on, but it remains in that condition, it forgets to switch off the mechanism, so then there will be um, the consequences will be very bad for that cell. Okay, so next is signal amplification. Uh, just like phosphorylation dephosphorylation process, some enzymes are also there which actually cascade the amplify cascade the enzyme those enzyme cascades can amplify the cellular response. At each step the number of activated products is much greater than in the preceding step. So by this way the signal is getting amplified. And in this example, you can see uh, that this is a very general phenomena of cell signaling, where the cell signaling molecule is bound to the receptor, and then the with the second messengers, the cell can generate the response. So this is a pathway. One single pathway leads to a single response. So this is a usual normal pathway. But sometimes the signal can be divided into two branches, and thus they can they can generate different responses inside the cell. And in another scenario it might be the case that one pathway is activated but another pathway is also helping towards that response process. So that means there is a crosstalk between these two pathways, signaling pathways. So you can see that this enzyme, this molecule is responsible for this response 4 here, but this is not activated until this molecule is present. That means two simultaneous signals are required for this response, and this kind of crosstalk in between two cellular pathways, signaling pathways, are also required. And you can understand that inside the cell these kind of crosstalks among the pathways are very much usual phenomena, and thus they can generate a huge network of cell signaling. And uh, the fourth one explain that different receptors can lead to a different response. So obviously that is very um, usual case that not only one single receptor but multiple receptors molecule are present on the cell, and they can generate its individual effect or individual responses. So at the last, the termination of the signal. So inactivation mechanisms are very much essential for cell signaling mechanism. If the ligand concentration fall, fewer receptors will be bound and unbound receptors revert to an inactive state. Thus, the termination can happen using different regulatory mechanisms and which is at most required for cell signaling mechanism. If the termination of the cell signaling is not operating well, so then the cell can be mm, going through the different pathways like apoptosis. So then, you know, uh, either that cell will enter to the apoptosis pathway, or the cell can go through the cancerous pathway. So anyone can happen next time. So both of these are in your syllabus. So next we will see what do we mean by apoptosis, and then we will see what is what do we mean by cancer in molecular biology called concept. Okay, so that's all for cell signaling mechanism.